Okay, so first we'll begin with the nucleosome protection assay. So nucleosome protection assay. And pretty much what this is, it's a laboratory method that's used in biochemistry as well as in genetics. In order to identify molecules of each RNA. That are present in different RNA samples. So RNA samples that has been extracted from a cell. Extracted from a cell. Okay. Alright, so this strategy is able to detect one or more RNA molecule in a known sequence at low levels. So, write that down. Detect one or more RNA molecules in a known level. Okay, so pretty much how we would draw this is we'll have our diagram here. Okay, and so we would have our labels. So this would be the DNA ligase, and this is its unit. So we would have it at 1.5. And in this case, we will be looking at the neurons that are present in the hypothalamus. So this area would represent the hypothalamus cell, and then this area here would represent the red blood cell. Okay, so pretty much you'd have your little indication for each one. Okay, and then here this would be DNA from neurons in PVK1 of hypothalamus. And then in this area here, this would represent the red blood cell. Okay, so pretty much, okay then. So for this scenario, we'll be looking at the DNA from the neurons in the hypothalamus. So in this case, while we're observing this, while using this technique, what would happen is that the DNA from the neurons would be degraded, while the red blood cell wouldn't be degraded. So you would still see a mark present here. And the reason for this is because the red blood cell is considered to be a condensed chromatin. So condensed chromatin. And pretty much when the chromatin is condensed, there is no transcription present. So no transcription. And then there also isn't any degradation. So degradation not present either. So hence why we still see the mark. But in the case of the DNA from the neurons, it is degraded. So that means that transcription is present and that this DNA is ready for gene expression. So ready for gene expression. Okay. So pretty much the reason for this is because when it comes to condensed chromatin, it's tightly packed and the histones aren't allowing the degradation to occur. So pretty much the histones are pretty much protecting the red blood cells. But as for this case, it's considered to be a decondensed chromatin. Oh, I forgot to write that down. So decondensed chromatin. And pretty much it's pretty much loose, so the DNase is gonna be able to chop this up because of how loose it is, and it's not really like tightly together. So quick kind of summary for nucleosome protection assay is, so we're looking for transcription if the chromatin is condensed. That would mean, chromatin is condensed, that would be no transcription, as well as no degradation. So no degradation. And then, oh, wait. Forgot something. So no degradation from DNase. And yeah. Okay, so we're looking for transcription. If chromatin is condensed, there's no transcription, no degradation from the DNase. Okay, now when it comes to looking at northern, it's a laboratory method, and it's used in order to detect certain RNA molecules within an RNA compound. And then it can also be used to analyze a RNA sample from a specific tissue or type of cell in order to measure the RNA expression of a certain gene. Okay. So another thing to note is that this method is able to analyze by size. So analyze by size, as well as the stability level of a particular RNA which is present in a complex sample. Okay, so pretty much in short, the RNA is sized by gel electrophoresis and is transmitted by melting into a membrane. Where the RNA is bound together. Okay, so now that we got a little kind of summary from Northern, okay, so let's draw our little diagram. So we would have our little box here, and we'll use the same scenario as in the nucleosome protection essay, but in this case, instead of it being the DNA from the neurons, it would be the RNA from the neurons that's present in the hypothalamus. And I probably spelled that wrong, but okay. And then this side here would represent the red blood cells. And so the time frame that we'll be looking at it is at a 48 hours. Okay. So pretty much we would have our mark right here. So in both. But instead of like in nucleosome protection assay, when the mark was right here on this side, so go back, instead of it being present here in the red blood cell area and not being present here in the neuron side of the diagram, it'd actually be reversed. So there would be a mark right here when it comes to the RNA from the neurons, while there won't be a mark present for the red blood cells. So the reason why is because we're pretty much looking for the presence of RNA as time passes. So, in other words, the more RNA that's present, then that would pretty much equal to more transcription to occur if gene is present. So in this case here, we can see that there's more RNA when it comes to the neurons, hence why the mark here after the 48-hour mark in comparison to the red blood cells. So that means that there's more RNA of the neurons than there is of the red blood cells. So here, mRNA levels are being compared in a, we just use this abbreviation RBC for red blood cell, in a red blood cell, a neuron from the hypothalamus. So, as we can see, as the extraction was from the hypothalamus, then it makes sense for there to be more RNA that's present for the neurons in the hypothalamus than there are of red blood cells. So again, hence why the mark is here and the mark is absent when it comes to the red blood cells. But yeah, thank you for watching.